Welcome to the Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. We're so glad you are joining us for this deep dive. And before we jump into the amazing science today, please do take a moment to like, subscribe, and maybe share this with your friends and family. Your support, it really is the engine that keeps the Science Experience running. We appreciate it. Okay, so today we are zooming right in on something fundamental, really, protein regulation. Specifically, we're tackling, well, one of those really tough, persistent questions in cell biology. How does a complex tissue, like in mammals, made of all these different cell types, how does it actually manage its protein machinery?
The proteome. Yeah, our mission here is to understand that balance, the dynamic quantitative balance between making protein synthesis and getting rid of the clearance. And critically, across individual cells inside a living system, not just an average.
Right, and that dynamic part, that's the huge challenge, isn't it? Because, historically, people mostly looked at mRNA levels, kind of assuming, more RNA means more protein, which isn't always true.
Exactly. Or they use cell lines grown in a dish, which, let's be honest, is pretty far removed from how cells actually behave in a real tissue.
Totally different environment.
Yeah. So trying to quantify the actual rates, synthesis, and clearance inside thousands of different single cells within a living tissue, that's been, well, technically almost impossible.
A real bottleneck.
A huge bottleneck. It means the actual in vivo story, the complex dynamics in life, has been, largely unexplored.
Until now, maybe. So the source material we're diving into today, it sounds like it tackles this head on.
It really does. This study, they looked at mouse trachea tissue, and it's just a landmark technical achievement, honestly. They systematically quantified the whole protein life cycle synthesis degradation across, get this, over 4,200 single cells.
Wow.
Yeah. And they managed to integrate single-cell proteomics with transcriptomics. So you get this full picture, this continuum gene expression regulation.
That sounds incredibly powerful for us trying to learn how this works. Okay, let's unpack that. Measuring protein quantity in one cell is hard enough, but the rates, turnover. How on earth do you track a protein breaking down in real time inside something so tiny?
Yeah, it required a really clever mix of technologies. The core idea is something called metabolic pulse labeling. Basically, they fed the mice a special diet. It contained lysine and amino acid, but a heavy version. It had extra neutrons, so it weighs slightly more, plus 6 Dalton.
Heavy lysine.
Okay. Right. It's harmless, but when the cell builds new proteins, it uses this heavy lysine. So think of it like a date stamp almost. All the proteins made during that feeding window get this heavy tag.
Ah, so you can tell the new ones from the old ones.
Exactly. The old existing proteins are light, the new ones are heavy. Then the challenge was reading that information from thousands of individual cells all at once. And for that, they combined the labeling with a high-throughput single-cell mass spectrometry method. It's called plexDIA. And they use something called mTRAQ barcoding, too. These are really precise analytical chemistry tools.
Okay, so labeling tells you new versus old, and the mass spec lets you measure it cell by cell.
You got it. The short version is this whole setup let them separate, identify, and quantify the heavy, newly made proteins versus the light, older ones in every single cell. And by measuring that heavy to light ratio over time, they could actually calculate the synthesis rate and the degradation rate for about 5,506 different proteins.
Across 4,200 cells, that's just an astonishing amount of quantitative data.
It really is. It gives you the protein half-life in its natural home, basically.
Okay, but I remember reading they hit a snag, or rather, something they had to be really careful about. Something about amino acid recycling. Why was that so important?
Ah, yes. Absolutely vital. And the fact they tackled it is, frankly, a huge validation of their whole approach. You see, amino acid recycling is a known issue when you try to measure turnover in vivo. When a cell breaks down an old protein, it doesn't just check out the amino acids. Often, it reuses them.
Makes sense. Waste not, want not.
Right. But if it reuses that tagged heavy lysine to build a new protein, well, that new protein now looks older or more stable than it actually is.
Oh, I see. It messes up your timestamp.
Exactly. It artificially inflates the half-life you calculate. So the researchers, they had to explicitly model this recycling rate and correct for it. And interestingly, they found it wasn't the same for all cells. Epithelial cells, immune cells, these seem to recycle more.
Huh. Cell type specific recycling.
Yeah. And the impact of correcting for it was huge. Before they applied this correction, the average protein half-life they calculated was around 7 days.
7 days, okay.
But after they put in the rigorous cell-type specific correction, it dropped. down to about 3 days.
Wow, that's more than double the speed, from 7 days to 3.
Massive difference. It means without that careful modeling, our whole picture of how fast things turn over in a cell was significantly off, skewed. Proteins are actually being made and broken down much faster than we might have thought based on older methods. And that 3-day average, it really backs up the accuracy of their in vivo measurements.
Okay, so they got the accurate measurements, they overcame the technical hurdles. This brings us to the big picture, right? Did they just end up with a massive list of protein half-lives, or did they find something deeper, like an organizing principle?
They did. And it's actually quite elegant, quite simple at its core. They found that the main way a cell regulates its proteins, whether it focuses on making them or clearing them out, is linearly dependent on how fast that cell is growing.
The growth rate dictates the strategy.
Precisely. The pace at which the cell divides seems to set the primary mode of regulation for its entire proteome.
Okay, let's break that down, because that sounds like the key takeaway. How does growth rate connect to making versus clearing?
Right. So think about how a cell gets rid of proteins. There are 2 main ways. One is active degradation. The cell literally uses machinery to chew up the protein. The other is passive dilution. Every time the cell divides, the existing proteins get split between the 2 daughter cells, so the concentration effectively halves.
Okay, active breakdown versus just getting diluted by growth.
Exactly. Now, if a cell is growing very slowly, or maybe not dividing at all, that dilution rate is really low, close to 0.
Right.
So to manage its proteome, that cell has to rely almost entirely on active degradation. Protein clearance, that's what they call the clearance rule.
Slow growth means active clearance is king.
You got it.
Mhm.
And they saw this clearly. In the slowest growing cells, they looked at these cartilage cells, chondrocytes, in the trachea, which didn't divide at all during the experiment. Active protein clearance explains something like 45% of the variation in protein levels.
So nearly half the control comes from just breaking stuff down.
Yeah. Degradation is the main regulatory dial for stability and function in those slow cells, sort of decay-driven stability.
Okay, so what about the other end of the spectrum, the fast dividers, like those basal stem cells you mentioned?
That flips the whole equation. That's the translation rule. As the growth rate goes up, that passive dilution from cell division becomes a much bigger part of the total protein clearance.
Because the cell is constantly having its contents anyway.
Right. The cell is effectively clearing out half its proteins just by dividing. So active degradation doesn't need to work quite as hard or have such a wide range to control things.
Ah, so the regulatory burden shifts.
It shifts towards controlling production. The influence of translational regulation, how fast the cell actually builds new proteins, that increases in direct proportion to the growth rate. So for those faster growing basal cells, active clearance only explained about 29% of the variation. Control shifted more towards translation.
It's like a resource management choice. Why waste energy actively degrading if dilution's doing a lot of the cleanup for you?
Exactly. It's efficient.
So, okay, we have this universal framework. Slow cells use clearance, fast cells use translation. But is it that simple? Within that is the regulation specific. Does every single protein follow the same pattern in, say, a chondrocyte versus a basal cell?
No, it's definitely not that simple. The regulation is still highly customized. While the growth rate kind of sets the overall stage, the fate of any individual protein still depends on the protein itself and the specific cell type it's in. It's about function.
Okay, so layers of control.
Right. For example, they found some proteins that seem to be regulated pretty much the same way everywhere. There was an enzyme, SALT1D1, involved in metabolism. It seemed to be controlled mainly by RNA levels, by transcription, in all the cell types they looked at.
Okay.
But then another protein, SEC14I2, involved in moving lipids around. That one was regulated mostly by translation speed, how fast it was made. Again, across different cells.
So some proteins have a preferred mechanism, but what about cases where the cell switches the mechanism depending on context? Can the same protein be controlled differently in a slow cell versus a fast cell?
Absolutely. And that really shows the sophistication here. The example they give with a protein called S100A1 is perfect for this. S100A1 does different things in different cells. In those slow-growing chondrocytes, the cartilage cells, they seem to need more of it, maybe for structure, maybe signaling. How do they get higher levels? Well, they boost the RNA transcription and they actively slow down its clearance. They basically turn down the degradation dial to hold on to it longer.
Okay, make more, break down less. Makes sense for a slow cell needing stability.
Right. But now look at the faster growing basal cells. There, S100A1 levels are lower. How do they achieve that? Primarily by suppressing translation, They just don't make it as quickly.
So the fast cell uses the other lever, the production lever, to achieve the opposite outcome for the same protein.
Exactly. It highlights the cell's genius, really. It doesn't just stick to one mechanism. It picks the lever transcription, translation, or clearance that makes the most sense given its growth rate and what it needs that protein for. It's about efficiency and function.
That's incredibly dynamic. Beyond individual proteins, though, didn't the study also find something really striking about how whole pathways or functional groups are coordinated? Something about protein correlations versus mRNA correlation?
Oh, absolutely. This was, I think, one of the most profound findings for understanding how cells actually work. It really shows you where the functional coordination is happening. They found that correlations between different proteins, how their levels go up and down together, were way stronger than the correlations between their corresponding mRNAs.
Way stronger. Like how much stronger?
Significantly. They found over 5,000 pairs of proteins that had a meaningful correlation, say above 0.3. But when they looked at the mRNA for those same pairs, only about 340 pairs showed that level of correlation.
Wow, that's a huge difference. More than tenfold.
Oh, huge. What it means is that looking at protein levels captures this deep functional coordination that you often completely miss if you only look at RNA. And they checked these strongly correlated proteins. They often belong to known functional groups or protein complexes, like those listed in the Quorum database. Proteins that work together are regulated together at the protein level.
Even if their RNA levels aren't tightly linked.
Exactly. Even if they aren't physically stuck together in a complex, if they're part of the same pathway, their protein amounts are coordinated.
So what's driving that coordination at the protein level, if not just synchronized transcription? What ties these functional groups together?
Well, according to their analysis, a significant part of that coordination comes from protein clearance regulation, active degradation.
Ah, so breaking things down helps keep things synchronized. That seems counterintuitive.
Yeah, but think about it like this. Active clearance allows the cell to fine-tune the relative amounts, the stoichiometry, of proteins within a functional pathway. Transcription might get you roughly the right amounts, but degradation provides that precise post-transcriptional control to ensure everything is perfectly balanced.
Okay, I see. Like quality control and balancing the team.
Exactly. Take their example of glycolysis in those chondrocyte cells. Remember, chondrocytes live in low oxygen conditions, so they rely heavily on glycolysis for energy. The study suggests that the coordinated clearance of the different enzymes in the glycolysis pathway helps keep that entire functional module tightly balanced and efficient. It ensures the whole metabolic team is present in the right ratios, ready to go.
A level of control that the upstream mRNA data just wouldn't show you.
Precisely. The cell uses degradation strategically to manage functional units.
This really does shift the perspective, doesn't it? It feels like this study provides a much more complete, dynamic view of gene expression.
It absolutely does. These findings, they reveal this quantifiable, organizing principle growth rate, dictating the balance between translation and clearance in a complex living tissue.
It takes us way beyond just a static snapshot of RNA or protein levels. We're seeing the life cycle, the resource management in action.
Right. And it provides the tools and, importantly, the conceptual framework to understand how cells manage themselves under normal conditions.
And presumably, this framework also gives us new ways to think about what goes wrong in disease.
Definitely. If this growth-dependent balance is the norm, you can immediately start asking, okay, what happens in cancer, where cells are forced into abnormally high growth rates?
Does the regulation break down? Does it switch entirely to translational control?
Or what about aging? We know protein degradation machinery can become less efficient with age. How does that disrupt this balance in different cell types?
This approach lets you quantitatively pinpoint where those disruptions might be happening. That's powerful.
Incredibly powerful.
Well, this has been absolutely fascinating, a real deep dive into the hidden life of proteins. I think that's probably all the time we have for today. Thank you so much for walking us through that.
My pleasure. It's truly groundbreaking work.
It really sounds like it. And thank you, our listeners, for joining us. Please remember to like, subscribe, and share this deep dive with your network. It genuinely helps support the SANS Science Experience and lets us keep bringing you these kinds of insights. Until next time, keep exploring the amazing dynamic world inside the cell.